I'm glad to have this opportunity from the A-CURE committee to present my analysis of extracellular proteins involved in the overloaded and unloaded heart after myocardial infarction. My disclosures are none. All right, so a little bit of background. We know that acute left ventricular unloading by percutaneous left ventricular assist devices, or PLVADs, benefits the compromised heart in a variety of ways. We know that unloading the LV into the aorta will reduce workload and reduce myocardial oxygen consumption. We know that there is reduced LV filling pressures and LV volumes, which leads to reduced wall stress. And we also know that pumping into the aorta will increase coronary perfusion. And my colleague will also touch about how later on how reduced wall stress can also improve coronary perfusion. But what if there's more to this story? What if there is another mechanism that underlies the benefits of PLVAD utilization? There have been previous studies that have examined the effects of unloading at the intracellular level. There have been groups that have studied calcium handling and mitochondrial dynamics. However, there has not been any published studies so far looking at the extracellular matrix. So, what is the extracellular matrix? The cardiac extracellular matrix is a dynamic scaffold of macromolecules that surrounds the cell. It's able to transmit the forces that act upon the heart, and it also responds to these forces and changes itself in response and signals to the cardiomyocytes in response to force. So, and through this, it modulates a number of processes such as cell signaling, cell adhesion, angiogenesis, and fibrosis. So, we hypothesized in our study that the change in the composition of the left ventricular ECM proteins is another mechanism that underlies the benefits provided to the heart by acute unloading, LV unloading with PLVAD. So to do this, we took 10 Yorkshire pigs and on day 0 induced MI using a balloon catheter that was inflated in the mid-LAD for 90 minutes and then deflated. 1 week later, we then split the groups into 2 groups, N of 5, with half of them being unloaded for 2 hours with an Impella, and the other half having acute AR mechanically introduced to cause an overloaded state. So after 2 hours, we sacrificed the pigs and then isolated the ECM from a remote site on the left ventricle. The ECM that was isolated was then denatured into ECM peptides, and then we screened these peptides using liquid chromatography and mass spectroscopy with the Sequest library for identification of these ECM proteins. And then this data was subjected to computer analysis. When we looked at the proteins that we got back from our analysis, we were able to identify a total of 986 proteins in our ECM isolates. From this number, we took these proteins and then plotted them by their significance value and their fold change value. We used a cutoff of a fold change of either greater than or less 50%, or a downregulation of more than 50%, and we use a p-significance value of 0.05. And from this, we were able to identify 39 differentially expressed proteins in the ECM of these hearts. With these 39 proteins, we then we did gene ontology analysis to understand this a little bit better. Gene ontology analysis is used to sort gene products and properties and relate them to each other. And there's three domains that are covered by gene ontology. There's the cellular component, or where can you find this product. We didn't look too closely at this, as we've already enriched our isolates for ECM. There's also molecular function, which is what it does. What does this protein do in the cell? And then there's biological function process, which is why does it do its function? So when we did the analysis by molecular function, we found on these 39 proteins, we found that there was a favoring of RNA binding and lipid binding proteins. And then when we looked at them by biological process, we saw that the processes favored were cell differentiation, vesicle transport, and programmed cell death processes. So in summary, we saw that there were significant differences in the protein composition of the cardiac ECM between the acutely overloaded and unloaded myocardium, and that these differentially expressed proteins tend to be involved in the molecular functions of RNA binding and lipid binding, and biological processes of cell differentiation, vesicle transport, and programmed cell death. Now, this suggests that there's a possible initiation of changes in cardiac cell phenotype in response to the unloading process, and it is possible that these changes induced by the P-LVAD unloading can have benefits that continue past device removal. Because when you unload the LV with the P-LVAD, we saw that the ECM responds, and through this response, we saw that there's upregulation, an increased amount of RNA binding and lipid binding proteins, and that these proteins can then potentially cause a change in protein synthesis for select cellular processes, and then that this might be leading to the beneficial changes that we see in cell phenotype during unloading. Okay. So future directions, I think that we would like to further characterize the effects of the differentially expressed proteins on the cardiac tissue, and also see if this can lead to identification of potential therapeutic targets. What I thought was really interesting about this study is that was mentioned earlier that now there's a movement towards using Impella for diagnostic purposes, but what about also for discovery purposes as well? So I'd like to thank my team at the Ishikawa Laboratory for whom this work could not have been possible. I would like to also thank Dr. Mayer for doing the sequence analysis for us. I'd like to thank the Center for Comparative Medicine and Surgery at Mt Sinai, and I'd also like to extend a great thanks to A-CURE for this opportunity. So thank you so much.